Welcome back, children. Now, till recombinant DNA combination now formed, whether it is through plasmid vector or through page vector or through any mode or Agrobacterium tumefaciens plasmid or retroviral plasmid, whatever the mode, now the recombinant DNA is ready. Now we need to inject or we need to introduce this recombinant DNA into the host cell. How can we achieve this introduction of recombinant DNA into the host cell, or we can call it as transformation of recombinant DNA into the host cell. Here, the recombinant DNA, now look at the slide, should enter cell to transform cells. But DNA cannot enter cells in a medium without treatment, because DNAs are called it as hydrophilic, that is, it is water loving, and it is negatively charged molecule. And it should enter means the cell membrane. Normally, if it is animal cell, cell wall is made up of cell membrane, and if it is a plant cell or bacterial or virus, they'll have a cell wall, and we need to treat the cell wall for the introduction of the your recombinant DNA. So the cell membrane is made up of bilipid protein layer. That is automatically this plasma membrane is hydrophobic, but your DNA is hydrophilic. So how this hydrophilic can enter through hydrophobic. So it is a little task only. So we need to, before introduction, we need to prepare our host to compete to enter the DNA into the host cell. So that is only mechanism behind competent host. Okay, so under that we have some techniques. We'll see what are the techniques. So host should be made competent to take up DNA containing vectors. So why we need to make competent means because nature of DNA is hydrophilic, but the plasma membrane is hydrophobic. So we need to make a host cell to receive the DNA containing vectors. And what are the techniques we'll use? We'll see. Now look at the slide. Okay, first one, chemical treatment. A treating host like bacteria with specific concentration of divalent cation. Divalent cation means calcium 2 plus, okay. That is only divalent cation. So we need to treat that is host bacteria with cation, divalent cation, that is example calcium 2 plus, and automatically it will increase the efficiency. It will increase the efficiency and it will help to increase the permeability of the cell membrane. It will increases the efficiency means what? It will increase the permeability of the cell membrane and make DNA enter through cell wall of host. And here, first method. And when it enters means they need to attach with that host. They need to enter the host and they need to go for replication other process. For that, another procedure, that is heat shock. Sudden change in temperature. First we need to cool and we need to increase the temperature, that is chill and hot, or we can call it as hot heat shock treatment. And here mixture of recombinant DNA is introduced into the host cell. And we need to incubate at 42 degrees Celsius temperature, okay. Incubation of host recombinant DNA, ice, then placing them at 42 degrees Celsius and then back to ice. So we need to continue this procedure for some times, and automatically it will enable bacteria to take up recombinant DNA. So this will make bacteria to receive the recombinant DNA containing plasmid or vector into the body of the bacteria. So what we want to do, first we need to add calcium 2 plus, that is a divalent cation like calcium, makes or increase the permeability of cell membrane to enter the DNA containing vector, and also we need to add we need to do ice plus heat treatment, and also to incubate for 42 degrees Celsius. It will enable to enter or to take the bacteria of the recombinant DNA containing vectors. Next we'll see here what are the methods we can use for the procedure under vectorless injection of recombinant DNA. So what we saw before is transformation of DNA through a vector. Okay, second part is a vectorless injection of DNA into the host. So under that actually we have four types. Under that we have four types. Okay, we'll see one by one quickly. So if imagine if you want to add your recombinant DNA directly into the nucleus of the host cell, and it can be done through a process called it as microinjection. Okay, that is common for animal cells. Say you write one example if you want. Imagine a human egg cell, and when we inject a sperm, automatically the sperm nucleus will fuses with the egg nucleus. So this is the example for microinjections. And recombinant DNA directly injected into likewise how the artificially sperms are injected to the egg, and how the fertilization takes place, like that only. It is not same. Like that, recombinant DNAs are directly injected into the host cell, so it will mixes with the nucleus, and it's automatically injured the nucleus, and automatically it started to form many copies. And third one, BioList or gene gun. And not this is microinjection is especially for animal cells. Microinjection is especially for animal cell. That is very, very important. Next, biolistics or gene gun. It is a host cell bombarded with a high velocity. Very, 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 very important. Host cell, and this is for plant cell only, children. Biolistic or gene gun. Here, host cell are bombarded with high velocity microparticles of gold or tungstens, and these microparticles are coated with a recombinant DNA, and it is used for plant cell. Imagine this is your recombinant DNA, and it was surrounded by a gold or tungsten material, and this material was, now it is called it as microparticles containing DNA, and the size should be normally 1 to micrometer. So if it is beyond that, we can't able to inject also. So we need to make microparticles. So with that, this is your gene of interest, and we are adding, surround that we are adding our gold or tungsten. So these together we can call it as microparticles. For an understanding, I have drawn big one, but it is actually very, very small, and it is what is the size? It is 1 to 2 micron. 1 to 2 micron only. 1 to 2 micron only. Clear? And automatically, and what is the size? Okay, size is 1 to 2 micron, and what speed it should go, and in that condition only we should regularize the timing and everything. With that only we can able to achieve. If not, it will reach plasma membrane, it will come back. If not, it won't cross even the cell wall. Sometime when it is more speed, it will get into the nucleus, and again it same speed will come out from the nucleus also. So there are so many practical problem is there. To overcome that, a constant rate was maintained, that is 1400 feet per second, SEC, 1400 feet or 1400 feet per second. Okay, here this much length, and this only the same seconds should follow to achieve the biolistic or gene can in the plant cell. Clear? Here three concept. Plant cell, it is used for plant cell. We are making recombinant DNA around, we are adding some gold or tungsten particles, so we can call it as microparticles. It should be 1 to 2 micron only, and we should follow some speed to achieve our target. If not, it will come out or it will not get inside also. So what is the speed? 1450 per second. By using this, we'll achieve biolistic or gene can. And the next procedure, we can call it as disarming pathogens. So pathogenicity of vector is removed, that is disarmed pathogen, and disarmed pathogen vector allowed to infect host or transfer recombinant DNA. That is only we saw the Agrobacterium tumefaciens. Apart from that, we have one more technique that we can call it as chemical. So what we saw before and all, with the how the vector will enter into the host cell, how it will multiply, and without vector, how can we introduce our gene of interest directly to the plant or animal cell. So example for animal cell is your microinjection. Here there is no vector. And example for a plant cell, no vector, it is a gene gun or biolistic. And if it is not, if it is will be in laboratory condition, so gene of interest will be, and automatically your gene of other interest or other protoplasm will be. So we used to keep one chemical that we can call it as PEG, polyethylene glycol. Okay, polyethylene glycol. This chemical will help to mix the recombinant DNA into the protoplasm of any plant or any animal cells or any microorganism cells, which will be in the laboratory. Clear, children? Microinjection for animals, plastic, biolistic or gene gun for plants. And if it is in a laboratory, so we need to mix a protoplasm of any cell or microorganisms or any part of plant cell or animal cell. So we used to keep the protoplasm, and with that we are adding gene of interest. So we need to keep one chemical, polyethylene glycol, will help to induce the fusion of both the gene of interest and the other protoplast. It might be protoplasm, might be bacterium, or it may be of any plant cell or animal cell, it might be anything. So that can be done. So that PEG, it comes under chemical method. So first method, microinjection. Second method, biolistic. And third, we can call it as the chemical method. Example, polyethylene glycol. And one more is there that we can call it as electroporation. Okay, fourth method is electroporation. In that, electric current is given, high voltage, short time. The electric current is given, automatically it will help to make some tiny pores in the host cell. This is temporary only, and it will help to enter our desired recombinant DNA into the host cell. And after, when we remove our high voltage, short time electrons, automatically it will close the pore of the cell membrane. So this is also another method in which vectorless gene of interest can be introduced into the host. Is that clear, children? So for vector, we learned, okay, how can be used the divalent cation, and vectorless category, chemical method, polyethylene glycol, and gene gun by using gene gun with the for plants, and microinjection for animals, and electroporation for any plants or animals. Only we are using electric current to make some temporary pores. Automatically the your gene of interest let into the host cell. Thank you, children. Next one. So this is the gene gun, children. This is the microinjection. We'll see first. Okay, this is a microinjection needle with foreign DNA, and it is a holding pipettes. Host cell will be, and its cell containing nucleus. Directly with the nucleus will add automatically the fusion of the nucleus of one cell with the injected cell, recombinant cells. So this is for mainly for animal cell. Okay, this is microinjection, and this is, we can call it as the next one we'll see. Okay, next one, remaining process of recombinant DNA technology, that I will explain in next class. Thank you, children.